Title of the talk is What Makes RNA Special? And the point I'm going to try to make is 3 points. And these are really the take-home message. RNAs form many interesting structures. Even though it's a single polypeptide, polynucleotide, thank you, Mike. It's a single polynucleotide that would be a single-stranded RNA. It can fold on itself and form a lot of very interesting structures that you're going to hear quite a bit about in this next few days. When it forms these structures, it can do many interesting things, and I'm going to outline a few of these for you. And probably the most intriguing thing for me, and I think for most of the people who have been working with RNA for several years, is that RNAs are always surprising us. It's always a lot of fun for us to pick up a journal, talk to colleagues, and so forth, and find out the latest thing that RNA is doing. So there are lots of surprises, and these surprises keep coming. It's not as if everything happened 20 years ago or 30 years ago. We're still getting surprises as we speak. And that's probably the most intriguing thing for me and for many of my colleagues. Now, what can RNA do? It can store information. That's pretty obvious. Messenger RNA. There's viral RNAs, and so forth. It can also, as you're going to hear, it can catalyze reactions. Ribozymes are the catalytic form of RNAs, and it can guide the expression and processing and stability of other RNA molecules. And as I said, there always are surprises. RNAs normally do not work solely on their own, but are very often collaborating with other molecules. And in particular, they collaborate with proteins. They bind proteins, and the proteins are actually very often an essential component of whatever function the RNA has, be it in degradation of a target or modification of a target, and so forth. So they're collaborators. They also recognize sequences by base pairing, and they have very often we'll see an RNA molecule will have functional domains. One end of the RNA, one part of the RNA will have one function, another part of the RNA will have another function, and we call that multitasking. And an example of multitasking is shown here, where you're trying to do everything at once, and you're not really designed to do it that way, okay? So this is one way you could do it, but it's not very efficient. There are ways that you can multitask and do it quite efficiently if the design is correct. And the kind of design that could happen is shown here, where you can have multiple functional domains that can each independently carry out an activity. And that's what RNAs generally do. So messenger RNAs, for example, have a coding region. They have a 5' and 3' 5' cap, poly A at the 3' end, and untranslated regions between the two ends. Transfer RNAs have multiple domains that have their own functions. The anticodon, these are just a few examples. The anticodon, and also the region that accepts the amino acid. We're going to hear quite a bit about ribosomal RNAs. There's peptidyl transferase activity, translocation, and decoding of the messenger RNA. And there are regions of the ribosomal RNAs that have been shown to carry out these individual functions. And likewise, what we call guide RNAs, molecules whose main function, as far as we know, is to bring proteins to targeted sequences in some other molecule, be it a messenger RNA or a precursor of a messenger RNA, or to some other target that the RNA is able to recognize. And examples that you'll be hearing about in this course are microRNAs, small nuclear RNAs, and long non-coding RNAs. So we'll be hearing about these as well. Now the different domains can be mapped out, and this is just an example of a ribosomal RNA where you'll hear a great deal more about this from Marat later today, where there are domains in the RNA that have specific functions. And these domains are formed by base pairing of the RNA as it folds back on itself. And so we have double duplexes here interrupted by single-stranded loops and bulges and so forth. And these are, they all contribute to the function of the RNA. This is the, I believe this is ribosomal RNA. There's a small molecule here. This is transfer RNA. And again, there are different domains in the transfer RNA that have specific functions. Anticodon at the bottom and the acceptor stem at the top. Now these structures are formed by this because of the sequence of the RNA itself. And with RNA, just with DNA, there's the canonical base pairing of AU or GC, but there are lots of non-canonical interactions as well that you'll hear about. There are stacking of bases, one on top of the other. There's some interactions to regions of the bases that one does not normally see in a normal duplex, as we're all taught about, say, with DNA. And a very important interaction is due to the fact that the RNA has a 2' hydroxyl group in the nucleotide. Okay, so that distinguishes it from DNA, of course. So this 2' hydroxyl group, I want to spend just a few minutes talking about it. The 2' hydroxyl group has the ability to hydrogen bond to lots of structures that DNA doesn't have that capacity because it doesn't have that hydroxyl group. This also changes the conformation of the sugar in the base, and that has implications in what kind of duplex the RNA can form. It's also responsible for the fact that RNA molecules are labile. They turn over quickly. They can be attacked by specific nucleases, but also it can engender sort of autolysis because the 2' hydroxyl can attack other regions of nearby phosphates. So that makes RNA labile, and as you'll hear from, I think from John Abelson in a few minutes, it plays a key role in messenger RNA splicing as well. There are other examples of the function, but these are just a few that I wanted to bring up. So what is this 2' hydroxyl? So here is just a schematic version of a nucleotide. The ribose, the 5' phosphate, 3' phosphate in the base, and the 2' hydroxyl group that we're talking about is this one down here. And that in DNA is just a hydrogen. And as I said, the 2' hydroxyl is responsible for hydrogen bonding, for enzymatic activity, attack on the ribose, and for conformation of the sugar. This is called sugar pucker. And just an example here is the form here that where we draw the ribose as sort of a little pentagon, and we tend to think of it as on a plane. But in fact, it's not a plane, it's puckered. It either goes like that or like that, depending on two different conformations. And with RNA, the 2' carbon in the sugar has a hydroxyl coming off in this direction here. And if the hydroxyl group is attached here, it's going to affect the stability or the energy of whether that 2' carbon wants to be up or down, as we're drawing it here. If it's down here, the way I look at it, facing down, it puts the hydroxyl group out, so it's not going to be interfering with interacting with other entities in the base or in the sugar. If it's up here, there's going to be more interference, and what one sees is that this is the preferred conformation for RNA. Okay, so RNA likes to have the sugar in this way. In DNA, where there's no hydroxyl group, this is actually the preferred version. Okay, the consequences is not just looking at the sugar, but there are consequences with regard to the distance between the phosphate bonds on either side of the sugar. And so you can see it goes in DNA, that you have 7 angstroms between those phosphates. With the RNA in this particular conformation called 3' endo, the distance between the phosphates is reduced by about an angstrom. Okay. Consequences of that is it's going to change the overall structure when you form a base pair between the bases here and some other base on another part of the chain. And that gives us the structures shown here. On the right-hand side, we have the normal B DNA that we're all B conformation of the duplex that you see with DNA. With RNA, because of this change here in the distance between the phosphates, you can see it's much more compressed. It's a little bit broader. And the grooves here changed. This is the major groove in DNA. This turns out to be the equivalent major groove in RNA, and it's very narrow. Even though we call it the major groove, it's the same faces of bases are in here, but they're very difficult to access. And so the broad part that can be accessed, if at all, is in the minor groove, what we call the minor groove of the duplex in RNA. And that's just because of that 2' hydroxyl group. As a consequence of the base pairing and the stacking and so forth, we have these very elegant structures, and this is obviously an old slide, and there's many, many more structures that we know about. That tRNA, this is one region from a ribozyme, self-splicing ribozyme, and here is a region you'll see a lot more about. That's from Marat in a ribosomal. And in the ribosome, you can see all of these elaborate structures that are important for the function. And I'm sure this is very much an outdated slide, but anyway, you'll hear about it more. So what can RNA do? Well, it can store and transfer information. Now, I'm going to go through each one of these very briefly and give you an overview. In storing information, we have messenger RNAs. As you all are aware of, taking the information from DNA sequences to to the ribosome. Viral genomes. Okay. One thing that we can think about, and I wonder if anybody has an answer here, is while we have viral genomes made out of RNA, we don't have, as far as I know, there's no cellular genomes made out of RNA. And anybody want to take a guess why that might be? Yeah. Yes?
At the end, the DNA is less labile because of this, actually, this old age.
Yeah. Yes, it's less stable. Yeah. Is that what you mean? Yeah, that's what the answer I want. Yes?
Because one thing is RNA is so much mutation prone and it have to be changed. But DNA is more stable than the RNA due to regarding this type of mutation. If all the cellular genome is replaced with RNA, and then the world of cells are going to be mutated and various types of cells are there.
Yeah.
Due to this type of changing of genome structure.
Right. Yeah. So it's really a matter of stability. And the fact that RNA is not stable can be an advantage in some ways because we can get RNA turnover, but it can be a disadvantage if you want to store the information for a long time. Yeah, that's right.
So when you're asking questions, the student, say your name first.
Okay, this young man here.
I'm Mike Garrison. You just electrified a part of my brain that was dark.
Is that possible?
By asking your question. We could actually change the genome to RNA, right? I mean,
Yeah, we could easily do that.
we could take some simple thing and change its genome to RNA and see if it could live. Just do it, so that you could switch it over to have no deoxynucleotides with the right polymerases and so on.
That's right. I mean, it's a good experiment to do. Yeah, yeah. Or one other way we could think about is go into a swamp and select for some sort of bugs that might survive where RNA has an advantage, like maybe in very acidic conditions or something like that. But I mean, that one might think about going along those lines. I mean, if in your experiment, we'd have to think about repair enzymes and the polymerases that would be necessary for it. But it's still, I think it'd be a fun experiment to do and see what this creature's like. Yes, in the back.
Louder.
Yeah.
I'm from Int'l from Slovenia. Actually, I wanted to just comment. I'm a microbiologist. That there are certain eukaryotic organisms that have part of their life cycle only as where their genome is only in an RNA stage. So they exist.
Yeah, so there are organisms that have RNA as a stage. I mean, viruses are a key example. You're talking about a cellular organism?
Yes. Actually some unicellular organisms.
Oh, that's very interesting. We should talk.
New. It's like a year, maybe half a year old.
Okay, we should talk about that with Yaris. I think that would really be fun. Okay. Good. This is the kind of discussion I think we should have throughout the course. All right? Okay, sorry. Changes in the environment. Say if in bacteria, if there's bacteria are lacking a particular amino acid, it'll change the conformation, ultimately conformation of the messenger RNA, which may change the ability of the cells to either translate that whole messenger RNA, or there will be a signal that will get back to the nucleus or to the DNA, to the chromosome, and affect translation, transcription of the gene. RNAs respond to changes in the environment, and that is if a messenger is not needed or is it's important to be degraded under certain conditions, it will be attacked, and oftentimes this attack is mediated by complexes that have RNAs in them, as you'll hear both from Donald and from Lynn, that there are specific mechanisms for turnover of messenger RNA. And this instability allows for flexibility, and it also is important that these messengers don't accumulate. I mean, if we kept making messengers and they never turned over, the cell would be so stuffed with messenger. So it's actually a good thing for the cell to be able to get rid of the old messengers and make some new ones. It's kind of like professors. You want to get the old professors out of the university so the young professor can come in and do their own thing. Several of the teachers in this course are facing this right now.
You'd like to leave the message behind in some way, and the information behind it some way that seems imperfect.
John says, if you want to leave the information and the message behind, what?
But if this messenger RNA is equivalent to a professor and you fire him,
Yeah, so you gotta...
what are they going to do after we're gone?
Yeah. Have a party. Another aspect of information transfer is alternative splicing and splicing in general. And we're going to hear quite a bit about that today, where you have one transcript that can be processed in alternate ways to end up with two different kinds of, or multiple different messenger RNAs as a result of splicing one pattern or the other. You end up with proteins that can be similar in one domain but different in other domains, and this is responsible for a huge increase in the complexity of the proteins that we can generate in our genome, from information in our genome. What can RNA do? Catalyze reactions, ribozymes. You'll hear quite a bit about splicing today, both messenger RNA, ribosomal RNA, and peptidyl transferase, which is the component that actually synthesizes proteins. Viruses replicate. Many RNA viruses replicate using a scheme where the product of the transcript actually can self-cleave and end up making a genome just by cleaving its own sequence in a specific place. And that's essential for the replication of a number of viruses. And as you'll hear from Mike Yaros, ideas about the origin of life require or fit in very well with the fact that RNA can both have information and replicate information, okay? And you'll hear Mike discuss that. What else can RNA do? It can act as a guide the expression or processing of other RNAs and the survival. I'll talk a little bit about guide RNAs. These are molecules whose main function, at least in the context that I'm discussing here, is to act as a guide to bring a protein to a target. Okay, it gives it some specificity to the protein's binding. And the microRNAs you'll hear about both from Donald O'Carroll and from myself later on. We'll talk about microRNAs, siRNAs, in terms of degrading or silencing messenger RNAs. The processing of pre-messenger RNAs requires small nuclear RNAs, and all these little prefixes here make a world of difference, obviously, in terms of what the RNA is doing. So small nuclear RNAs participate in splicing of pre-messenger RNAs. There's another class of small RNAs called nucleolar RNAs, small nucleolar RNAs, and they guide proteins that will direct methylation, modification of bases in other RNA molecules. A type of RNA that has recently come on the scene, it's been around for forever, but we're only aware of it quite recently, is something called CRISPR RNA. And Donald will talk about CRISPR RNA as a mechanism of defense of cells. This is another example of these surprises that come out of nowhere, and suddenly it's really the big thing. And then long non-coding RNAs. We'll hear some discussion of these. They are involved in chromatin activity in the nucleus, control of gene expression, control of stability of messenger RNAs, and organization of chromosomes as well. This is a sort of a catch-all term. It more describes the RNA rather than the function. It's big, it's more than 250 or so nucleotides. Non-coding, as far as we know, it doesn't make encode a protein, but it has a function. And this function can be anything. And I think if we're talking about unexpected surprises that are going to come out of wherever in the next few years in terms of what RNA does, I think these long non-coding RNAs are going to be responsible for a lot of it, because there's so much. We know these things exist, these RNAs. We know a few of the functions, but they don't. This function over here doesn't mean that that it's universal for all non-coding RNAs. Different molecules will have different functions, and we're just going to be discovering these functions as time passes very soon. This is just an example of how a guide RNA can work. It recognizes a sequence in a messenger RNA, recruits a protein, a specific protein that will bind to that guide RNA. Go sit down here on the messenger RNA, in this case in the 3' UTR, and that protein will recruit other proteins, which ultimately either silence the messenger RNA or lead to its degradation. And you'll be hearing about this later on. So what makes RNA special? Getting back to the first point, they form lots of interesting structures, do a lot of interesting things, and they're constantly surprising us. Now, a number of the people in this room have been around to see some of these surprises appear on the in front of us, and I'll just sort of, just off the top of my head, I just thought I would list some of these surprises that in my career have come along. First of all, there's splicing. I mean, we all take it for granted that there's splicing happens, but 30 years ago, that wasn't the case. Nobody thought about splicing, and that was a really strange event. How did you relate this long precursor to the messenger RNA or the tRNA? The fact that RNAs can act as guides was also a surprise. Ribozymes, that was a real surprise. People thought, oh, only enzymes, only proteins can catalyze reactions, which of course we know is not true. RNAs that can affect the activity of proteins and enzymes, either in polymerases or synthetases and so forth. The fact that RNA can be used in experiments to dissect possible events leading to the origin of life, and sort of molecular fossils, which we can see today. So I mentioned earlier this CRISPR RNA you'll hear about, which is a defense mechanism. The microRNAs, siRNAs, and so forth that will affect other messengers. 20 years ago, we had no idea that these things existed, let alone that they would function. And now these guys are really important in not only in our understanding of biology, but in the practice of medicine. I mean, they're prognostic indicators, diagnostic methods that depend on the presence or abundance of certain microRNAs. And we now know they are essential for sort of the rational practice of medicine, even. Epigenetics, control of chromosomic organization, and communication. And there, as you'll hear from Marvin Wickens, the complex networks within a cell can be coordinated by the use of specific RNAs. These are just some of the surprises that have come on the scene in the past few years, and I'm very excited. I think we should say we're all extremely fortunate to be doing biochemistry at this time, because there is so much that is just coming into view, and it's the excitement of this whole field that I think gets us out of bed every morning. So I thought I would give you a sort of an overview of some of the talks that are in this course here, and the way they illustrate what RNA is doing. The bulk of the time today will be spent on messenger RNAs, both how they're produced and how they function in terms of, say, encoding of proteins, and then how they are subject to proofreading. Lynn is going to talk about making sure that the way the cell knows what messengers are being made, and the correct ones. So that's today. Tomorrow we're going to talk about other things that RNA can do, other than just act as messenger RNAs. So I just classified this sort of in terms of human activities. We have little policemen and big policemen. The little policemen are the microRNAs, siRNAs, maybe the CRISPR RNAs that are sort of controlling things. Big policeman is a way of sort of, these are big molecules that control the stability of other messenger RNAs. So these are multiple different ways of affecting, in a very specific way, the activity of the survival of a messenger RNA. They're going to be translators. We're going to hear about tRNA, and communicators. Marv is going to talk about the way that different RNAs communicate with each other. They're philosophers, and that's Professor Yaris, in talking about origin of life, and so we'll all know where we came from, hopefully. And then what I call the discoverers, and the discoverers I think is really an important and exciting part of this course, which is we're going to have some presentations by some of the students, some of you out here, about discoveries that you're making right now in your own labs. And I think that is the way we're going to be, that's where the future lies, okay? And the current research on these are all listed in your program. And I haven't read the abstracts, but I did read obviously I read the titles of these talks, and they look really exciting. And I think we're going to have a good time tomorrow afternoon hearing these reports. On Wednesday, we'll talk about mechanisms of how RNA does what it's doing in terms of splicing and the origin of, more in the origin of life, and translation. Mara's going to talk about the ribosome function. And then more in terms of an organismal approach, Donal and Marv and I will talk about sort of cell biology as it is impacted by RNA. And then on Thursday, there will be a truncated session, but in that session there'll be discussion of RNA protein interactions. Okay, both in terms of tRNAs and chromatin. Okay, and there may be some modifications in the program as some of the teachers decide that they would like to switch the order of their talks or something like that. So with that in mind, don't be confused if the talk that is on this schedule differs from the talk that is actually delivered. I hope that the teachers will explain what they're doing. So let me just conclude by saying, yes, RNA is exciting. RNA does a lot of things, forms great structures that are really interesting on their own. These structures can carry out and perform a lot of functions for RNA and make a lot of interesting discoveries for all of you who in the room during your careers. So with that, I'll stop. Thank you.